Welcome to the Chronicles of Berberine, a biochemical tale loosely fashioned off the Chronicles of Narnia, the seminal work of C.S. Lewis and his colleague and personal physician R.H. Poling. The Chronicles take place in Narnia, a magical make-believe kingdom into which human children and open-minded adults can enter through the wardrobe and grow old and gain great wisdom within the kingdom of Narnia. And when the human children are old and gray in terms of Narnian years, they can be whisked back into the land of man and take with them all the knowledge they've accrued and use it to do good. Narnia was a wonderful kingdom to live in. The rocks, the brooks, the trees, the sky, and the stars were all animate and lived in peace and harmony with the good people of Narnia. The summers were long and cool. The harvest was full. The doctors did not have to do excessive paperwork. But not all was well in the kingdom of Narnia. A band of goblins and ghouls and evil elves, led by the vile Queen Jadis, sought to kill the human children and enslave the good people of Narnia, make it winter forever but never Christmas. She was going to force her subjects into Jadis care. Those that did not sign up were turned to stone, their physicians were emasculated. And as the story goes, a battle for the future of Narnia was fought, a battle between good and evil. The Narnians, led by the human children who were their generals, against the goblins and the ghouls and the other evil minions of Queen Jadis. The human children put on a brave face, but deep inside they were frightened because they know that evil is a formidable adversary. Evil is seductive with great rewards for bad behavior. To defeat evil, they knew they needed the deep magic of Narnia. They had to go back in time and understand why Queen Jadis was an evil witch. In centuries past, she was a good queen, and she presided over a harmonious and joyful kingdom. But over the years, over the centuries, there were episodes of strife and tribulation. The harvest was not successful. The food was unequally distributed. There was injustice and bigotry. And over the years, over the centuries, her view of humanity became narrowed and corrupted, so all she could see was evil. Well, the human children did what they needed to do and learned about the conditions from which evil can spring forth. And with this, they gained the deep magic and used it to conquer the evil queen and her minions and vanquish them from Narnia forever. And that leads us into the Chronicles of Berberine. Now, Lewis and Poling wrote this story in the 40s when the good people of the world had gotten together to recognize and conquer a great evil. But they're warning us it is not sufficient to deal with the end-stage manifestations of evil. If we don't deal with hunger and injustice and inequality, the conditions from which evil is springs and is nurtured, we will have to fight Queen Jadis again in the form of an Andropov, a Gaddafi, a Saddam, or a Putin. So I'm a physician, and I would like to give you a little bit of deep magic as to the origins of the evil that is compromising your physiology. And this brings us to the Chronicles of Berberine, and this is an adaptation of a 4-part lecture series that I give within the Fellowship in Anti-Aging and Regenerative Medicine. Now, question for you, what is the natural predator of man? Is it lions and tigers and bears? Oh my? Well, that would be the wrong answer. The natural predator of man is infection. The leading cause of death in the history of mankind is perinatal sepsis, infection during the birthing process. That is the number one killer. And if we make it into infancy, we are likely to die of a number of other microbial infections just before we reach the age of reproductive maturity. Gram-positive cocci like staph, gram-negative rods like E. coli, coliforms like Vibrio that causes cholera, rickettsia, plague, a host of viruses. I think we can say that bubonic plague altered the course of human history far more than did Henry VIII, Hitler, or Mussolini. We've been evolving on the planet for 3 million years. Our greatest threat is infection. We've gotten pretty good at recognizing and defending against infection. Now, my thesis, my position is that the decision to increase production of cholesterol, the reason your cholesterol is a lot higher at age 50 than it was at age 20, is that this is a maladaptive response of our body to what it perceives as infection. And when we have an infection, particularly a chronic infection, we need a lot of cholesterol because we're making new white cells to fight the infection. They need to be fed, so our blood sugar needs to rise. The white cells need bullets, free radicals, and inflammatory cytokines. Our arterial wall needs to activate, allowing these white cells to enter our tissues and fight infection. Hyperlipidemia is a maladaptive response of our body to perceived infection. When we begin to generate cholesterol, in this pathway, we are also upregulating the generation of inflammatory mediators because we need not just cholesterol, we need bullets. So as we follow this pathway to make cholesterol, we also activate an enzyme called Rho kinase that activates an enzyme called NADPH oxidase that makes superoxide free radical to turn our white cells into killing machines. Unfortunately, they're causing inflammation, hyperglycemia, hyperlipidemia, and oxidative stress. Now, a key mediator of inflammation is a molecule called ICBA kinase. Our cells have receptors, threat receptors on the cell membrane for bacterial cell wall, lipopolysaccharide, or free fatty acids, or cytokines, or angiotensin. When these receptors are ligated by a molecule that they perceive as threat, that sets in motion a chain reaction of serine kinases that eventuate in phosphorylating or activating an enzyme called ICBA kinase. Now, ICBA kinase, which we're going to personify as Queen Jadis, is a mediator of inflammation amplification. It will degrade the inhibitor of a molecule called nuclear factor kappa beta. Nuclear factor kappa beta is a transcription factor. It is held in abeyance in the cytoplasm, the cell fluid, by its inhibitor, ICBA. When Queen Jadis degrades the inhibitor, nuclear factor kappa beta can translocate in the nucleus and begin to change the way we read our genetic code to produce inflammation, oxidative stress, endothelial dysfunction, because our body thinks we're responding to an infection. Now, Queen Jadis, not only does she mediate inflammation, she mediates insulin insensitivity. She makes our blood sugar go up. A really good idea if you're fighting a chronic infection, but not a good idea when you're a modern man trying not to develop hyperlipidemia, hyperglycemia, and cardiovascular disease. Not only do we activate nuclear factor kappa beta, we upregulate a number of inflammatory pathways, such as activator protein 1. We're making superoxide free radical by activating NADPH oxidase, and we are downregulating expression of NRF2. That is a beneficial transcription factor which codes for reading of our antioxidant and our anti-toxicity defense mechanisms. But we don't want a lot of antioxidants when we're fighting infection. We simply want to make bullets to kill these microbes that we perceive are attacking us. Now, not only will inflammation lead to hyperlipidemia, when we are inflamed, we have a cholesterol sensing negative feedback system that prevents our cells from overfilling with cholesterol. This feedback mechanism is defeated with inflammation. Thus, our white cells and every cell of the body can engorge with cholesterol more irrespective of what your cholesterol level is. With inflammation, the liver becomes a cholesterol generator. Our white cells will engorge with cholesterol, and this promotes atherosclerosis and heart disease. In addition, we can oxidize our LDL molecules, and now they no longer look like food. They look like a bacteria, and they will be recognized by scavenger receptors of our white cells and incorporated. And this plays a key role in the initiation and propagation of cardiovascular disease. Now, I've been practicing cardiovascular medicine for 28 years, and I wasn't taught much about nuclear factor kappa beta or activator protein 1. I was taught that the primary focus of cardiology was on anatomy. There's a fixed blockage, a concretion, compromising the supply of oxygenated blood, rendering it insufficient to meet the needs of the heart muscle at rest or with effort. So my job was to do a coronary angiogram and determine the severity and extent of the narrowings, and then make recommendations on how we could best resupply oxygenated blood with bypass surgery or percutaneous interventions. If the pattern of blockage was not amenable to a surgical procedure, we would use drug therapy to decrease your heart rate and blood pressure, decrease demand. Cardiology was about supply, demand, and fix blockages. And of course, this is an appropriate approach to crisis. If you come in with a heart attack, we want to open up your blocked artery. But if we focus all of our time and attention and resources at dealing with high-grade fixed blockages and ignore the factors that cause this disease to develop in the first place, then we're going to spend all of our time in the hospital doing invasive procedures over and over again. We will not be effective at preventing this disease, which is the key. So dealing with high-grade blockages has given way to a study of the factors that cause cardiovascular disease, and we will call these risk factors. And we have a long list of cardiovascular risk factors, and we keep adding to it as we learn more. And as we'll see, berberine is effective in dealing with a number of these risk factors. Now, my approach to medicine is integrative. I describe myself as a practitioner of integrative cardiology and metabolic medicine. I'm board certified and university trained in internal medicine and cardiology. I'm now an advanced fellow in anti-aging and regenerative medicine. My colleague Stephen Sinatra and I wrote a book, a patient manual on integrative cardiology called Reverse Heart Disease Now. My practice is involved in research. We were involved in researching new nutritional approaches, new device approaches, and new drug approaches to cardiovascular disease. I've done 3,000 heart catheterizations. 20 years ago, I was tied with two older physicians as the leading admitter of cardiology patients to my primary hospital. Now I think I might be known as the leading non-admitter to the hospital, and I'm quite happy with that designation, because if I deal with the cause of the disease, I don't have to deal with the disease itself. Now, and in the Chronicles of Berberine, we're going to be discussing the use of berberine in dealing with hyperlipidemia, hyperglycemia, inflammation, and heart disease. But I'm also going to take this opportunity to teach you as much as I can about the physiology that underlies your health. Now, berberine is a well-characterized molecule. Technically, it's called a quaternary protoberberine type alkaloid with a dibenzoquinolizidone ring or something like that. It's bright yellow. It's obtained in a powder form. It's found in the rhizomes, which is the root structure, stem, and the bark of medicinal plants. The human dose is typically 350 to 1,500 milligrams per day. It is a non-toxic substance. It's very difficult to kill an animal with berberine. I think it's impossible to hurt a human with berberine. Now, berberine is a component of a number of medicinal plants such as Coptis chinensis French. In traditional Chinese medicine, atherosclerosis is associated with phlegm, blood stasis, and toxin accumulation. Chinese herbs that clear heat and detox may inhibit atherosclerosis via anti-inflammatory and immunodepressive actions. Coptis chinensis is felt within the framework of traditional Chinese medicine to clear heat and dry dampness, purging fire to eliminate toxin, and that's better than resorting to bypass surgery. Now, berberine is used in traditional Chinese medicine and in traditional medicine throughout the world. Some of the Chinese preparations are huanlang, huangbai, and umbelatine. Barberry is used throughout the world. We'll present a study showing how barberry extract is useful in controlling blood sugar and lipids in Iranians. Tree turmeric, Oregon grapefruit are Northern American medicinal plants that contain berberine. Now, huanlang, it was discussed 2,000 years ago in traditional Chinese texts. In Note of Elite Physicians by Hong Jingtao in 500 AD, its ability to lower blood sugar was discussed. Shenong's Herbal, which was published 200 years ago, described the use of Coptis to treat bacterial dysentery and infectious diarrhea. Its use in diabetes became known in 1988. There was a patient who was diabetic who presented with bacterial dysentery, and berberine has antimicrobial action. It's been traditionally used to treat a bacterial infection of the gut. And in this individual, their blood sugar fell, and that led to study of the use of berberine in treating hyperglycemia. Berberine entered the modern scientific age when Dr. Kong showed that berberine will upregulate the expression of the LDL receptor on cells of every cell in the body. And now in 2014, I've been studying berberine for 2 years now, and I think it's of universal value in the prevention and treatment of cardiovascular disease, and I think that everyone should take that. And when I made that statement, my colleagues in the fellowship said, well, gee, if you think berberine's of universal value, you must show that berberine meets Pohling's postulates. Now, we all took biology in high school. We learned about Koch's postulates. These are the rules. These are the provings that you must show if you want to show that an infectious agent causes a disease. Well, Pohling looks kind of like Koch. You know, he's a fairly old man now. He looks a lot younger than he is. That's because he works with me. I'm an anti-aging physician. But these are Pohling's postulates for a treatment that is of universal value in the prevention and treatment of cardiovascular disease. First of all, it must deal with atherosclerotic risk factors and the causes of metabolic syndrome and type 2 diabetes, insulin insensitivity, overweight, hyperlipidemia, hypertension. It must improve endothelial function, lower oxidative stress. It must quiet down inflammation without damping your appropriate immune defenses, preferably with an antimicrobial effect, and it needs to blunt collateral damage of inflammatory reactions. Its mechanisms of action must be well defined at the levels of transcription and translation. It must be low in cost and associated with low toxicity. Ideally, it will synergize with pharmaceutical and device therapies, and there must be published studies to document mechanisms and efficacy. So I'm going to have to prove Pohling's postulates for berberine. Now, the key physiologic effect of berberine is to upregulate an enzyme called AMPK, AMP-sensitive protein kinase. Now, ATP, adenosine triphosphate, is the currency of energy exchange within our body. It's adenine, ribose, and 3-phosphate groups. We need ATP to do work, and we need high levels of ATP to drive forward all useful chemical reactions in our body. Vibrant health is associated with high levels of ATP. As ATP falls, one by one, cellular functional mechanisms become depressed. Now, ATP is a renewable energy resource. We have work to be done. We split ATP into adenosine diphosphate and a free phosphate group. We burn some sugar and fat. We tack on the phosphate group. We can make more ATP. We can recycle it. But when oxygen's not available or the caloric intake is insufficient, we will do work, but we have no energy. So we have ADP. We can combine two ADPs to make ATP and AMP, but inevitably AMP builds up. A rising AMP level, and particularly a rise in the ratio of AMP to ATP, means we have energy insufficiency, so we're going to activate AMPK. Now, AMPK, AMP-sensitive protein kinase, think of this as the enzyme of primitive man. It is activated when we need energy and we have experienced excessive oxidative stress. Consider primitive man would go on long treks following a herd of animals trying to obtain food. Primitive man would suffer from energy deficiency, and he would be generating a lot of free radicals in the long trek. Speaking of primitive man, here is me finishing a 26-mile marathon. And of course, I have a big smile for the camera, but deep inside, I feel awful. I'm suffering from energy insufficiency and free radical stress. So when I'm running a marathon, I'm also activating AMP-sensitive protein kinase, the enzyme of primitive man, that sends a signal to my physiology: don't build things, burn things. We need energy now, and do so in a fashion that decreases free radical generation. Berberine, caloric restriction, exercise, leptin and adiponectin, the prescription drug metformin and berberine, along with exercise, will activate AMP-sensitive protein kinase. And again, it sends a burn, do not build signal. Don't waste energy building cholesterol and triglycerides. Burn them. Slow down the rate of cell division. And as we'll see, this is helpful in preventing cell damage in response to toxins or preventing damaged cells from turning malignant. So caloric restriction, exercise, and berberine will upregulate this key enzyme. Conversely, inflammatory cytokines and caloric excess will downregulate it. And this is the condition of modern Western man. We're overfed, we're overweight, we're inflamed, and we are downregulating AMP-sensitive protein kinase, so we're getting hyperlipidemia, hyperglycemia. We're at increased risk of malignancy. We have endothelial dysfunction. We're out of balance, and we need to fix this. And we can fix this with lifestyle change and with berberine. Now, so when we activate AMP-sensitive protein kinase, we will physiologically downregulate HMG-CoA reductase. That is the target drug, target of statins. Statins poison that enzyme. Berberine turns it off. It becomes less necessary, so we make less cholesterol. As we will discuss, berberine stabilizes the messenger RNA of the LDL receptor. It blocks transcription of PCSK9, which degrades the LDL receptor. We make less cholesterol. We express more LDL receptors. We're going to lower our serum cholesterol. Via phosphorylation, we're going to inactivate an enzyme called ACC carboxylase, and this will promote the burning of fatty acids and lowering their production, and will lower our triglycerides level. We're going to increase the expression of insulin receptors on our cell membranes. We will become more insulin sensitive, and the signaling pathways within the cell that mediate insulin activity are upregulated. We are going to block insulin resistance, insulin insensitivity due to inflammation, such as bacterial cell wall, lipopolysaccharides, cytokines, saturated fatty acids. And we will provide physiologic protection against the toxic complications of diabetes, diabetic eye disease, neuropathy, kidney disease, and diabetic cardiomyopathy. We're going to have a favorable effect on weight physiology. We're going to have a larger number of small fat cells that are less metabolically active. This will help protect us from overweight. We alter the composition of the bacteria within our gut into a more favorable mix, such as that of primitive man. We will absorb fewer calories from carbohydrate. We're less likely to have leaky gut. We are going to have less leakage of bacterial lipopolysaccharide into our circulation. We're going to turn off NADPH oxidase, which is which generates the lion's share of free radicals within our body and within our artery wall. We're going to activate an enzyme called uncoupling protein, which helps with weight control and lowers the generation of free radicals. The deleterious transcription factors, nuclear factor, kappa beta, and activator protein 1, their translocation is blunted, while NRF2, which codes for antioxidant anti-toxicity enzymes, will be upregulated. This will protect us from inflammatory tissue damage and promote apoptosis, suicide of malignant cells. Endothelial function improves. Endothelial function, the ability of the artery wall to generate nitric oxide. This provides the Teflon coating to our circulation. Anything that improves endothelial function will retard the initiation and progression of arterial disease. Proper endothelial function will protect us from adverse cardiovascular events. Berberine upregulates endothelial function. Cardiovascular physiology improves, and we will discuss these characteristics of berberine in the section on berberine and cardiovascular disease. And the clinical effects, LDL and triglycerides decrease with minimal change in HDL. In italics, in bold print are the properties of berberine that I can prove with human studies. In italics are the beneficial effects that I'm going to extrapolate from animal studies. So in humans, berberine will lower LDL and triglycerides, and hyperlipidemia due to inflammatory stress is attenuated, extrapolating from animal studies. Glucose falls, insulin sensitivity improves, hyperglycemia in response to inflammation is blunted, and there's protection against the consequences of long-standing diabetes. Weight loss occur, it will help with fatty liver, protection against cell damage from infection, chronic inflammation, ischemia, that's insufficient oxygen, or toxicity. Berberine should protect against malignancy. In humans, tolerance to radiation therapy for lung cancers is enhanced. Blood pressure may fall, endothelial function improves. We protect the heart against stress and strain. There's protection against restenosis, at least in animal models. In humans undergoing stent placement for acute coronary syndrome, if we add berberine to standard drug therapy, we see lower cytokine elaboration, and improved clinical outcome. When we add berberine to standard drug therapies in humans with heart failure, we see an increase in functional status, improvements in ejection fraction, decreased mortality, lower arrhythmia, and berberine synergizes with and adds to our standard pharmaceutical measures. It's a very effective molecule in integrative cardiology and integrative medicine in general. And now we'll move on to the next section, which discusses berberine in cholesterol metabolism. The first book in the Chronicles of Berberine series is titled The Lion, the Witch, and the Wardrobe. So our section covering the use of berberine in lipid management will be titled PCSK9, AMPK, and the LDL receptor. We'll discuss the role of berberine in dealing with cholesterol, triglycerides, modified and oxidized LDL cholesterol, and in blunting inappropriate cholesterol esterification. I'm going to start with the clinical trial of berberine in low-risk individuals. We have 137 overtly healthy Italian men and women who are enrolled in Berlusconi care. And things aren't going so well in Italy right now. Their economy is in shambles, so they need cost-effective approaches. And these individuals were not at high risk for cardiovascular disease, but they had risk factors predisposing them to cardiovascular disease down the road. Their cholesterol was elevated between 200 and 240. They were overweight. Their BMI, upper limit of normal, is 25, so they were overweight to obese. They did not have high blood pressure. They were not smokers. And the researchers knew that risk for an initial or secondary repeat cardiovascular adverse event falls by 20% for every millimole reduction in LDL. That would be a 38 milligram per deciliter drop in LDL or total cholesterol. Now, drug therapy may not be cost effective here, and drug therapy, as we know, has side effects. So the question, will nutritional supplementation get the job done at lower cost and with lower risk? So what they did, they took these individuals and put them on a program of regular exercise and dietary modification over 6 months. This was the run-in period. Then after 6 months of diet and exercise, they randomized them to 3 months of run-in protocol and placebo, or run-in protocol with berberine, 500 milligrams twice a day. After 3 months, therapy was stopped. This was a washout period for 2 months, and then they crossed the subjects over to the opposite regimen. So each subject served as their own control. Diet and exercise and placebo, diet and exercise plus berberine, 1,000 milligrams daily. Now, with the 6-month run-in period, diet and exercise, you can see weight fell, body mass index fell slightly, blood sugar fell just 2 points, LDL fell 5 points, triglycerides fell. Those are favorable, but they're not large changes. Here we have our weight. The weight falls from 166.5 to 159 pounds with 6 months of diet and exercise. If we add in berberine, we lose 4 more pounds. During the washout period, there was no weight regain, and then back on berberine, the weight loss was maintained. Likewise, body mass index falls just a little bit with 6 months of diet and exercise. It falls more with berberine, and this is maintained. Fasting blood sugar falls slightly with diet and exercise. No major change with berberine, but off berberine it rises. You go back on berberine, it falls. Triglyceride falls a lot more with berberine. That's lost with washout. You go back on berberine, triglycerides fall. LDL cholesterol falls slightly with diet and exercise. It falls a lot more, from 157 to 133 with berberine. You lose that with washout. You go back on berberine, it falls. So favorable effects on triglycerides, LDL, and fasting glucose. Total cholesterol falls significantly with berberine. Now, that 33 milligram per deciliter reduction in cholesterol is just below our goal of 38. So simply via cholesterol reduction alone, berberine decreases cardiovascular event risk by nearly 20%. But that doesn't consider the additional risk reduction associated with the drops in triglycerides and glucose. And as we'll see, berberine improves endothelial function and augments our antioxidant defenses. So we have a low cost, basically a $30 a month supplement that provides effective protection against cardiovascular risk factors in individuals who are at low immediate risk, but relatively high long-term risk of cardiovascular disease. So let's talk a little bit about cholesterol and future mortality. We vilified cholesterol as the cause of cardiovascular disease, but is that actually correct? Because we know, conversely, that the risk of cerebral hemorrhage that's bleeding to your brain is inversely related to your serum cholesterol. That is, the lower your cholesterol, the greater is your risk for cerebral hemorrhage. This was noted in Japan in 1971 and confirmed in the US. In congestive heart failure, which is an inflammatory condition associated with oxidative stress, the lower your cholesterol, the worse is your outlook. Death rate, if your cholesterol is below 172, is twice that as individual with a cholesterol above 235. So what is it about high cholesterol that's protective in heart failure? Well, in heart failure, there's inflammation, there's bowel wall edema, bacteria translocate from the bowel into the blood, and cholesterol will neutralize that bacterial lipopolysaccharide. So cholesterol is not all bad for us. Now, with this understanding that cholesterol may be sort of a double-edged sword, in 1992, the National Heart, Lung, and Blood Institute sponsored a study. They analyzed 19 prospective cohort studies. Here, we take a large group of people, look at their characteristics, and then follow them over time and correlate the level of cholesterol or blood sugar or blood pressure with the risk of a future event. And what they did here, they looked at cardiovascular death rate, overall death rate, death-related non-cardiovascular diseases. They censored early deaths because if you had a undiagnosed cancer, your cholesterol might be low. So individuals that died in the first year were censored. Otherwise, they followed them for 9 to 30 years. They determined the optimal range for cholesterol with respect to mortality is 160 to 99. So now we're going to calculate long-term mortality risk relative to the optimal range of low cholesterol below 160, elevated 200 to 239, and markedly elevated above 240 versus optimal 160 to 199. And you see these curves. Now this is total mortality. This button here, these top two curves are men. And with men you see a bell-shaped curve. Total mortality increases with a high cholesterol. It's optimal between 160 and 200, and it also increases again with low cholesterol. The green dot here refers to women, and you can see there's no bell-shaped curve. There is no increased death risk associated with a high cholesterol. If anything, low cholesterol is associated with increased overall mortality in women. Now, if we look at cardiovascular disease, it's a little bit different. The top two curves are men. Yes, the greater your cholesterol, the greater is your risk of dying of cardiovascular disease, but this curve is damped in women. High cholesterol is not associated with a markedly increased cardiovascular risk in women as it is in men. But if we exclude cardiovascular mortality, if we look at death from respiratory tract dysfunction, digestive tract dysfunction, trauma, suicide, cancer, we see that the lower your cholesterol, the greater is your risk. High cholesterol is associated with a decreased risk of dying of cancer and other non-cardiovascular conditions. So maybe cholesterol is not that bad a thing after all. Maybe it's the way our body is managing cholesterol. Now, our body needs 800 milligrams of cholesterol. And if we don't take it from diet, our liver's going to make it. We need cholesterol to make new cell membranes, such as if we're making a lot of white cells to fight infection, to make steroid hormones, to make bile acids, to make vitamin D. So cholesterol is very important to the body, and its production and metabolism is tightly regulated. Now, cholesterol obtained from the diet or made in the liver is a waxy, fatty substance. It's water insoluble. So the body will bind it to a protein, apolipoprotein B100, to make a blood-soluble particle. That's the LDL cholesterol. Its job is to ferry cholesterol from the liver to meet the needs of the body. Excessive cholesterol can be stored within our cells as lipid droplets. This involves cholesterol esterification. As we'll see, this is upregulated in the presence of inflammation. We can also return excessive or used up cholesterol from the periphery to the liver via the HDL particle, and we have many strategies designed to upregulate HDL function. But the key controller of the serum cholesterol is the presence of the LDL receptor on the cell membrane of the liver. So we're going to talk about hepatic, referring to liver, cholesterol metabolism. Now, the LDL receptor is a molecular hand that sticks out in the circulation. It'll grab an LDL and it'll bring it into the liver cell and it'll break it down. Generation and expression of the LDL receptor in the liver is directly related to the liver's need for cholesterol. When the liver needs cholesterol, we're going to make LDL receptors, we're going to pull it out of the blood. When the liver has plenty of cholesterol, it'll stop making LDL receptors, we won't pull it out of the blood. So if we are treating you and wish to lower your serum cholesterol, all of our efforts are designed to increase the presence of the LDL receptor in the liver. Now let's talk a little bit about enzyme function. Our body is run by enzymes, and HMG-CoA reductase is the initial and rate-limiting step in the generation of cholesterol. It's the rate-limiting step in the generation of cholesterol, and because we're upregulating generation of cholesterol in response to what our body perceives as infection, it will also increase oxidative inflammatory mediators, free radicals and cytokines. Statin drugs work by blocking this enzyme. They block the generation of cholesterol and they turn off the faucet of inflammatory mediators. Now, when we activate HMG-CoA reductase, we're going to activate Rho kinase and we're going to activate NADPH oxidase. We're going to make a lot of superoxide free radicals. This inflammation will cause evil Queen Jadis to translocate nuclear factor kappa beta in the nucleus to transcribe a multitude of inflammatory mediators. Activator protein 1 enters the nucleus and does the same, and we're going to make more toll-like receptors. We're going to initiate a feedback loop of progressive inflammation and oxidative stress that leads to hyperlipidemia, hyperglycemia, endothelial dysfunction, and cardiovascular disease. What we call the pleiotropic beneficial effects of statins simply have to do with turning off this key inflammatory pathway, blocking upregulation of free radical generation by NADPH oxidase. We talk about immune modulation with statins. They simply follow blocking the generation of oxidative and inflammatory mediators. And we will give you statins to prevent you from developing arterial disease and converting your stable plaque to an unstable ruptured plaque, because this is all due to inflammation. It's all due to our body's misperception of our current milieu as chronic infection. Now, how do we control HMG-CoA reductase? Moment-to-moment activation and inactivation has to do with phosphorylation. If we phosphorylate HMG-CoA reductase, we turn it off and we mark it for degradation, so we'll break it down quickly. Long-term levels have to do with transcription and translation, how we interpret and read our genetic code. Now, AMP-sensitive protein kinase is a protein kinase. It'll take a phosphate group from ATP, bind it, phosphorylate it to a protein that will turn it on or off. Activated or phosphorylated AMPK phosphorylates HMG-CoA reductase, and that inactivates it. It doesn't poison it like a statin, it just turns it off because the body doesn't need it. And phosphorylated HMG-CoA reductase is turned off, so we are not overproducing cholesterol and inflammatory mediators, and it's marked for degradation. We're going to break it down. Transcription and translation. We have our double-stranded DNA, we open it up, and we copy it. This is called transcription, and we make messenger RNA, a template. The messenger RNA leaves the nucleus into the cell fluid, the cytoplasm, where the code is translated into the production of proteins. So we'll discuss how berberine influences transcription and translation in an anti-inflammatory favorable fashion. So here we have our double-stranded DNA. We're going to open it up, and a transcription factor will bind to a start button. Within the DNA, it's called a promoter, and read the DNA and then this will, then the messenger RNA enters the cell fluid where it's translated into protein. And we can influence transcription and translation with berberine. So these transcription factors, they're held in abeyance. They're held in jail in the cell fluid. When they're activated, they translocate to the nucleus to code for different proteins. Nuclear factor kappa beta and activator protein 1 are our response to perceived infection. And of course, that is, in modern man, that is the response to perceived infection with belly fat, leaky gut, gingivitis, and toxins. Conversely, NERF2, when it translocates to the nucleus, generates antioxidant and anti-toxicity enzyme system. So in my approach to medicine, I want to block nuclear factor kappa beta and activator protein 1 translocation and enhance NRF2 translocation. I don't need drugs and surgery if we can control your physiology and influence in a favorable fashion. Now, let's talk about liver hepatic cholesterol metabolism. SCAP, SRE binding protein, and sterol regulatory element interaction. Sterol regulatory element is the promoter site for transcription of the LDL receptor. So when a transcription factor, sterol regulatory element binding protein, binds to the SRE, that starts the reading of the DNA to make messenger RNA, which will be translated to make LDL receptors. SCAP is sterol regulatory element binding protein, cleavage activating protein. It is a conditional chaperone for the SREBP. So what we have here, SCAP and its partner in SIG bind to SREBP, the LDL transcription factor, and keep it basically in jail in an area of the cell called the endoplasmic reticulum. When there's plenty of cholesterol in the cell, SCAP will sequester SREBP. So we're going to stop making LDL receptors because we got plenty of cholesterol. If we're getting plenty from diet or if we're manufacturing it, we don't need more cholesterol, we don't need more LDL receptors. However, if the content of cholesterol in the cell is insufficient because we haven't had enough to eat, for example, SCAP will sense the low cholesterol. It will then translocate SREBP to another area of the cell called Golgi apparatus, where SREBP is modified, it's cleaved, it enters the nucleus, binds to the sterol regulatory element, and we start making LDL receptors. So we have this negative feedback system. When there's plenty of cholesterol in the cell, we won't make the LDL receptor. When cholesterol levels are insufficient, we will make LDL receptors so the liver can pull cholesterol in to meet its needs. So we make these LDL receptors. They are extended from the cell membrane of the liver cells. They will grab LDL cholesterol out of the blood. So we need to remember that the generation and expression of the LDL receptor is related to the liver's need or perceived need for free cholesterol. So when the sterol regulatory element binding protein binds its promoter, we're going to make more LDL receptors. But we're also going to make more HMG-CoA reductase. Now, HMG-CoA reductase makes cholesterol. So, and this, of course, makes sense. If the liver doesn't have enough cholesterol, typically because diet has been insufficient, we want to make LDL receptors to pull it out of the circulation, but we also want to make more cholesterol. So we're going to upregulate HMG-CoA reductase. And that sort of defeats us if we're trying to lower your cholesterol with drugs. And if the interaction between the sterol regulatory element binding protein and this promoter site also makes a protein called PCSK9, which stands for proprotein convertase subtilisin kexin type 9. So we're going to call it PCSK9. Now what PCSK9 does, it will bind to and degrade the LDL receptor. It will also be released into the circulation. If it binds an LDL receptor, it will break it down. So generation of PCSK9 will block the LDL receptor, and this will tend to raise your serum cholesterol. And this defeats us when we're using drugs to lower your cholesterol. Because we need to remember this system was not set up to make us good statin takers. It was good to prevent mankind from dying. Mankind would experience low liver cholesterol primarily due to starvation, limited cholesterol in the diet. So if we have insufficient cholesterol in our liver, we are going to make LDL receptors, but we're still going to start making more LDL, and we're going to make more PCSK9 that's going to break down the LDL receptor. And this will compromise our therapeutic efforts to lower your cholesterol. Because if we lower your cholesterol with drugs or with diet, yes, we'll make more LDL receptors, but we're going to make more HMG-CoA reductase that generates cholesterol. We're going to make more PCSK9 that breaks down the LDL receptor, compromising our therapeutic efforts. Now, PCSK9 is under genetic control. Gain of function SNPs, that single nucleotide interposition defects, subtle changes in the genetic code that make PCSK9 more effective, This is associated, this is one of the causes of familial hyperlipidemia. People that have a cholesterol of 500 or 600 because they're making too much PCSK9. And PCSK9 will be generated whenever there's insufficient cholesterol in the liver. Decreased intake from inadequate diet, starvation, decreased absorption, such as if we treat you with plant stanols or ezetimibe, which is a prescription drug, its trade name is Zetia, that blocks cholesterol absorption, or whenever we block HMG-CoA reductase with a statin or our green statin-like agents, red yeast rice extract, polycosanol oil of bergamot, this will upregulate PCSK9 and thwart our efforts to lower your cholesterol. Conversely, PCSK9 downregulation, there are individuals who genetically have low function of PCSK9. They have low LDL cholesterol, reduced cardiovascular risk. The drug companies are making drugs to downregulate PCSK9. There's a monoclonal antibody that is available by injection. It's not out in clinical practice. It's still in clinical trial. But if you get PCSK9 monoclonal antibody, your cholesterol will fall for 2 weeks. Well, we don't need to inject monoclonal antibodies to lower PCSK9 because, as we'll see, berberine blocks transcription, blocks the generation of PCSK9, which is why berberine will synergize with all other forms of therapeutic cholesterol reduction. And we want to lower our cholesterol, we want to lower oxidative stress, we want to lower inflammation because we don't want foam cells to develop in our artery wall. These are white cells, monocyte-derived macrophages engorged with cholesterol. This is the initial manifestation of atherosclerosis. It's the driving force. High cholesterol with inflammation and oxidative stress are white cells engorged with cholesterol because they think they're fighting a battle against an infection, which is really a perceived infection. We get foam cells and atherosclerosis. Now, in theory, our white cells, our monocytes, cannot overfill with cholesterol. See, they have LDL receptors. They're going to be expressed only when they need cholesterol. But our SREBP, SCAP, SRE interaction deals only with native free cholesterol. Now, if the monocyte has too much cholesterol in it, it will stop making HMG-CoA reductase, and it will stop making the LDL receptor. So the monocyte should not be able to overfill with cholesterol because when it has too much cholesterol, it'll stop making it and it'll stop making LDL receptors. However, in the presence of inflammation, our negative feedback system is compromised. When we're inflamed, we begin to esterify cholesterol within our white cells and every cell in the body outside of the liver. We will esterify cholesterol into lipid droplets. Then the levels of free cholesterol in the cell will fall, and then we'll start making LDL receptors. And this is why we're seeing people with normal cholesterol having heart disease, because it's not the cholesterol, it's how the body manages the cholesterol. With inflammation, our cells, particularly our white cells, can overfill with cholesterol. When cholesterol is oxidized by free radicals, such as superoxide, the LDL is degraded and it begins to look to the immune system like it's a bacteria. And it will no longer be internalized by the LDL receptor, but rather by the scavenger receptor, which misrecognizes the oxidized LDL as a microbe. Hyperlipidemia, inflammation, oxidative stress lead to atherosclerosis. Now, this system was designed to protect primitive man from its natural predator over the last 3 million years. The problem is the milieu of modern Western man is misperceived by our body as another infection. Visceral fat is inflammatory. Fat cells in the belly fat are infiltrated with white cells that think this is infected tissue. They make inflammatory mediators that drain to the liver. The liver's seeing chronic infection, leaky gut, toxicity, smoking, the inflammation from sleep apnea. Any sort of chronic infection will upregulate these pathways. Think about gingivitis. It's just your gums are inflamed, but as the severity of your gingivitis increases, so will your systemic C-reactive protein in the blood, a marker of immune system dysregulation, and we see your cholesterol going up. So simply chronic inflammation in the mouth leads to inflammation and hyperlipidemia. Any chronic infection, any chronic condition associated with oxidative stress and inflammation, the body says there's another infection. Our high sugar, our high fat diet, the toxins in our food, it's another infection. So we're responding to this perceived chronic infection with oxidative and inflammatory stress, hyperglycemia, hyperlipidemia, endothelial dysfunction, and now we're seeing heart disease in 35-year-olds. When I trained, if you came to the hospital with chest pain and you weren't 50 years old, we wouldn't even talk to you. And we weren't missing anything because young people didn't have heart disease. Now we're seeing 30-year-olds with heart disease because of the inflammatory nature of our lifestyle. Now, again, this misdecision to respond to perceived infection to generate cholesterol also generates free radicals. We translocate Queen Jadis. Queen Jadis will translocate nuclear factor kappa in the nucleus. Activator protein 1 translocates the nucleus. They code for inflammatory mediators, and we block NRF2 transcription because we don't need antioxidant and antitoxicity defenses. We want to spend all of our time making bullets to kill what we perceive as infection. Now, the way into Narnia is through the wardrobe. To get deeper into Narnia and gain the deep magic, we need to follow the path by the lamppost and learn about AMP-sensitive protein kinase, which we'll abbreviate as AMPK. Now, let's talk about berberine and how it upregulates AMP-sensitive protein kinase. We're going to take muscle cells in tissue culture. We're going to incubate them with vehicle. Those are placebo conditions with an artificial AMPK activator, a drug called idomycin or berberine. And we're going to measure AMPK and activated phosphorylated AMPK. We're going to measure ACC carboxylase that generates triglycerides and phosphorylated, which is inactivated ACC. So here you can see vehicle does not phosphorylate AMPK. Ionomycin, which is an artificial AMPK activator, does. Berberine activates it the most. So we're showing that berberine activates AMPK. Now, let's now look at rat muscle mitochondria. The mitochondria, the organelles within our cells where we burn sugar and fat to generate energy. And there is a biochemical chain reaction, complex 1 through 5, through which we generate ATP energy. And here we're going to measure oxygen consumption with the complex 1 substrate pyruvate. It is blunted by berberine. No effect on the other complexes. So what berberine is doing, it's sort of tricking the mitochondria by blocking energy generation, but not enough to create a net reduction in energy, but it's tricking the body. So what you're going to do, you're going to burn sugar anaerobically. This is called glycolysis. And we're going to get a transient increase in the ratio of AMP to ATP. So we're tricking the body into thinking that we are suffering from an energy deficiency, such as primitive man on a long trek, or me at the end of a marathon. So we're going to upregulate AMPK and send a burn, do not build signal throughout the body. Let's look at this in an animal model. Male Syrian golden hamsters, they're all going to be put on a bad diet, a high-fat diet, high cholesterol, coconut oil diet for 2 weeks. Then you're going to treat them for an additional 2 weeks with a vehicle. That's the control group, or vehicle plus berberine, 100 milligrams per kilogram. Then obtain labs and examine their livers. And what you see with berberine, LDL cholesterol falls, no change in HDL, hepatic lipids, levels of cholesterol and triglycerides fall within the liver, and hepatic fatty acids fall because you're making less cholesterol, less triglycerides, you're making fewer fatty acids. That's all desirable. Now we're going to take HepG2 cells. These are immortalized liver cells they're liver cancer cells that are immortalized. They are used commonly in cell culture research. We're going to take human HepG2 cells, incubate them with radiolabeled carbon over 6 hours with berberine at varying concentrations or vehicle, a control substance. We're going to measure incorporation of the radiolabeled carbon into cholesterol and triglycerides within the cells and secretion of the radiolabeled cholesterol and triglycerides into the cell media. Will berberine blunt lipid synthesis? Yes, it does. In a dose-related fashion, generation of cholesterol and triglycerides is blunted, less is secreted across the cell membrane. So we're blocking generation of cholesterol and triglycerides within these cells with berberine activating AMP sensitive protein kinase. Now here you can see we're blunting the generation of cholesterol. If we repeat the study with a molecule called PD-98059 that blocks an enzyme pathway called extracellular related kinase, berberine doesn't work. So berberine depends upon extracellular related kinase. I'm going to explain that more in a moment. Same thing with triglycerides. If we look at incorporation of carbon into acetate, that's another marker of lipid synthesis. It is blunted with berberine, and it's blunted by AI-CAR, which is an artificial AMPK. And this is more evidence that berberine is working by activating AMPK. So by activating AMPK, we block generation of cholesterol and triglycerides and glycerol synthesis. So berberine activates extracellular related kinase 1,2 that activates AMPK via phosphorylation. AMPK, when it's activated in turn, will downregulate HMG-CoA reductase. So we make less cholesterol. We're not poisoning the enzyme, we're just turning it off via Mother Nature's pathways, decreased LDL synthesis. We downregulate ACC carboxylase, decreased fatty acid and triglyceride synthesis. Berberine upregulates extracellular related kinase 1,2 that will, and we'll talk about this in a moment, will stabilize the messenger RNA for LDL. We make less cholesterol, we're going to make more LDL receptors, we're going to lower serum cholesterol. Berberine physiologically downregulates cholesterol and triglyceride biosynthesis via activation of AMP-sensitive protein kinase, and my guys are happy with this. Berberine and LDL receptor messenger RNA dynamics. We're going to take hamsters and feed them regular or a high-fat, high-cholesterol chow for 2 weeks. Obtain baseline labs, then we're going to treat these hamsters that are on a bad diet with vehicle, that's the control group, low-dose or high-dose berberine. We're going to evaluate lipids and liver histology, its appearance under the microscope at day 10. And transcription is reading our genetic code. Translation is translating the messenger RNA into the activated protein, into the enzymes. So here we see with a high-fat, high-cholesterol diet, serum cholesterol rises. Bad diet with berberine in a dose-related fashion lowers cholesterol. If we look at levels of LDL receptors on the cell membrane, and we call 1 unit on the high-fat, high-cholesterol diet, we make more LDL receptors. We make more LDL receptor messenger RNA with berberine. Now, in a high-cholesterol diet, the liver has plenty of cholesterol. It's not supposed to be making LDL receptors. But bad diet plus berberine, you can see an upregulation in the transcription of the messenger RNA for the LDL receptor. And this works because berberine phosphorylates extracellular related kinase 1,2, and that's associated with an increase in transcription or expression of the LDL receptor. Now, this is the best article published to date on berberine. This is Dr. Kong's work. He was looking at the biochemical effects of herbs used in traditional Chinese medicine. So he took extracts of 70 herbs and incubated them with the HepG2 liver cells in cholesterol depleted or cholesterol replete medium, and he found that of all the herbs that he studied, berberine was the most effective in increasing transcription of the LDL receptor. So he's looking at LDL receptor messenger RNA expression. Berberine was the most effective in a time and dose dependent fashion. Now, if you're cholesterol deficient and we give you berberine, you'll transcribe more messenger RNA for the LDL receptor. If you got plenty of cholesterol, that would tend to suppress transcription of the LDL receptor. But even when there was plenty of cholesterol, you can see an uptick with berberine. And so you make more messenger RNA. This is translated to make LDL receptor protein, which then appears on the cell surface. So we're increasing reading of the genetic code to make more LDL receptors, and they function on the cell membrane. Thus, they are going to lower serum cholesterol. GAPDH is a housekeeping protein. It's sort of an internal control. You can see that in a dose-related fashion, transcription of the messenger RNA for the LDL receptor is increased with berberine. This is in HepG2 cells, and they studied it in other liver cells. And then they also looked to make sure that these LDL receptors would work. Would they take up LDL? And indeed they did. How does berberine upregulate expression of the LDL receptor? Remember, when there's insufficient cholesterol in the diet, SCAP will take SREBP, it will activate it, put it into the nucleus, so we're going to make more LDL receptors. Low cholesterol is sensed by SCAP. The SCAP SREBP translocates from the endoplasmic reticulum to the Golgi apparatus. SREBP is modified, it's released from jail, it translocates to the nucleus. SREBP binds to the promoter site of the steroid regulatory element on our DNA, and we start cranking out messenger RNA for the LDL receptor, which is then translated into the LDL receptor protein that is then expressed on the cell membrane. So we're going to treat the HepG2 cells with berberine with and without lovastatin. Lovastatin is a statin. It is a metabolic poison. It blocks the activity of HMG-CoA reductase. So what you see with lovastatin in a dose-related fashion, we're going to make more messenger RNA for the LDL receptor. That makes sense. If we block the generation of cholesterol in the cell, SCAP senses that, it takes SREBP into the nucleus, we're going to make more messenger RNA for the LDL receptor. Berberine works just as well, and berberine will synergize with lovastatin. So the question comes up, is berberine a statin? Well, statins have a lot of benefits. They have side effects. We want benefits without side effects, and the answer is no. Berberine does not poison HMG-CoA reductase. It turns it off physiologically. It is not a statin. Well, if it's not a statin, how does it work? Could berberine lead to translocation of SREBP into the nucleus? Could it be an artificial SCAP? Well, let's examine this. A compound called GW707 will split SREBP from its chaperone SCAP. The SREBP translocates to the nucleus, binds to the promoter, and makes more LDL receptor messenger RNA. And you can see that GW707 translocates SREBP from the cell fluid, the cytoplasm, to the nucleus. Berberine does not. So berberine does not directly transport SREBP into the nucleus. It is not an artificial SCAP. So we've determined how it doesn't work. We need to determine how it does work. Could berberine be an artificial SREBP? Could berberine bind to the LDL promoter steroid regulatory element and read the genetic code itself? Is it a transcription factor? We're going to treat the HepG2 cells with berberine or the SREBP agonist GW707. GW707 will split SREBP from SCAP. SREBP translocates in the nucleus. SREBP binds to the steroid regulatory element binding site on the LDL gene promoter region, and we're going to make more messenger RNA. And berberine doesn't do that. Berberine does not bind to the DNA and initiate the transcription of the LDL receptor messenger RNA. So berberine is not directly inhibiting HMG reductase. It's not a statin. It's not blocking cholesterol synthesis pharmaceutically. It is not translocating steroid receptor element binding protein to the nucleus. It's not directly stimulating the steroid regulatory element within the LDL gene promoter region. So berberine is not directly increasing transcription of messenger RNA for the LDL receptor. But berberine does increase LDL receptor messenger RNA expression, LDL receptor protein expression, LDL receptor mediated LDL uptake. So how's this possible? We're going to take HepG2 cells and we're going to treat them with actinomycin, which will block transcription of DNA. So you stopped reading your genetic code. We do this with and without berberine. And under control situations, you will see a rapid drop-off in the level of messenger RNA for the LDL receptor because you can no longer make it because of the actinomycin. But the levels of the messenger RNA for the LDL receptor are prolonged in the presence of berberine. Berberine stabilizes the messenger RNA for the LDL receptor. The LDL receptor messenger RNA contains non-translated regions that mediate rapid turnover of the LDL receptor messenger RNA. This allows us to fine-tune LDL receptor expression. Berberine interacts with the non-translated region. It stabilizes the messenger RNA and prolongs its half-life, so it lasts longer giving it greater opportunity to be translated into LDL receptor protein. More LDL receptor protein will be translated, increased LDL receptor protein expression, your serum LDL will be lower. This is how berberine works, and this is why it synergizes with any other methodology that lowers cholesterol. All of our methodologies lower intracellular cholesterol, we make more LDL receptor messenger RNA. Berberine will stabilize that messenger RNA for the LDL receptor. You're going to make more LDL receptors. Berberine activates the extracellular related kinase 1,2 pathway, which stabilizes the messenger RNA for the LDL receptor. UO126 inhibits ERK1/2, and you can see it blunts the effects of berberine in upregulating the expression of messenger RNA of the LDL receptor. So with berberine, we're going to see more LDL receptors in the cell membrane. And we'll talk about this more in the next section. We're also going to see more insulin receptors on the cell membrane. As you treat with berberine, you'll see a dose and time related and synergistic increase in expression of the LDL receptor and the insulin receptor. We want lots of LDL receptors to pull cholesterol out of our arteries into the blood and back to the liver. We want enhanced expression of the insulin receptor to keep our blood sugar under control to prevent insulin insensitivity and diabetes. Berberine does both. Drugs don't do this. Statin drugs will lower your cholesterol and they raise your blood sugar. Many of the drugs we use to lower your blood sugar cause weight gain and secondary hyperlipidemia. Berberine will lower cholesterol, triglycerides, and blood sugar, in part by increasing expression of the LDL receptor and the insulin receptor on the membrane of every cell in your body. Now, we've talked about the biology. I understand this was tedious, but the more you understand about how your body works, the better, because in the final analysis, you are responsible for your health. You need to know how your body works. You need to know how your drugs and your supplements work and how they interact. So now we've talked about the biology. Let's talk about some more clinical trials. Berberine for lipid control in the elderly. 80 elderly non-diabetic hyperlipidemic subjects who were intolerant to statins or refused to take them. And these are a group of people that I would see in my cardiology practice. Age above 75, cholesterol above 200 or the LDL above 160, 60% were hypertensive, 25% were smokers. This was secondary prevention, 84%, 45% had known coronary disease, 33% had peripheral vascular disease. We're going to do baseline studies and randomize them to receive red yeast rice extract 200 milligrams a day, which is a green statin, and the therapeutic dose is 2,400 milligrams. This is a low dose. Polycosanol, which is also sort of a green statin, and berberine, which will physiologically downregulate HMG-CoA reductase, and of greater importance, stabilize the messenger RNA for the LDL receptor. And they use some low doses of CoQ, folate, and astaxanthin, an antioxidant. So you got the nutritional cocktail of red yeast rice, polycosanol, berberine, or placebo. You're going to repeat the baseline measurements over the ensuing year. With placebo therapy, there's no change in cholesterol, but with the nutritional cocktail, the cholesterol falls by 50 points. Now, a 20% reduction, 38 milligrams per deciliter, decreases your risk by 20%. So simply by cholesterol reduction alone, we're probably decreasing risk by 30%. LDL does not change with placebo therapy. It drops 40 points with the nutritional cocktail. HDL rises with both groups. Triglycerides falls significantly with the nutritional cocktail. Fasting sugar does not really change with placebo. It falls 5 points with the nutritional therapy. Hemoglobin A1C, which is a 3-month running average of your blood sugar, falls with nutritional therapy. HOMA, which is a measure of insulin insensitivity, falls. So, and this is because we're knocking Queen Jadis on her side. Queen Jadis, which activates nuclear factor kappa beta, not only does she mediate inflammation and oxidative stress and hyperlipidemia, she mediates insulin insensitivity. Because when you have an infection, you want insulin insensitivity, you want a high sugar, but we don't want that for our patients. So we are going to neutralize Queen Jadis and downregulate inflammatory pathways, lowering cholesterol, lowering triglycerides, lowering glucose, blunting inflammation. So clinical improvement, cholesterol falls by 20%, LDL by 31%, HDL rises by 10%, triglycerides fall by 10%, insulin sensitivity improves, and it was safe. There were no adverse effects on liver chemistries or CPK, that's muscle enzymes. When we give you statin drugs, we often see a rise in your liver chemistries. We see muscle inflammation, a high CPK. That doesn't occur with berberine. Mild side effects in 10% of the treatment group, 18% of the placebo patients. So treatment was associated with fewer side effects than placebo therapy. No events of mortality in either group. Berberine statin synergy. This is the really cool part, I think. 63 hyperlipidemic subjects who are not undergoing lipid-lowering therapy. Do baseline measurements, randomize them to receive over 2 months berberine, 500 milligrams twice a day, the standard dose, simvastatin, a statin drug, 20 milligrams a day, or the combination. Then we're going to repeat the lab studies. You can see that simvastatin and berberine both lowered your cholesterol. Berberine may have done a little bit better. Combination therapy, there was a synergy, dropping cholesterol from 260 to 184. Both simvastatin and berberine lowered LDL, but there was a synergy, dropping LDL from 168 to 115 with combination therapy. Triglycerides don't fall that much with simvastatin. They fall a little bit. They fall more with berberine, synergy here. HDL doesn't really change significantly with either therapy, and we wouldn't expect that. Now, HepG2 cells, these immortalized liver cells in tissue culture, we're going to incubate them with berberine or simvastatin and look at LDL receptor messenger RNA. So with simvastatin, in a dose-related fashion, we're going to see an uptick in LDL receptor messenger RNA. Lower intracellular cholesterol, SCAP releases SREBP, it enters the nucleus, it's going to make LDL receptors. That makes sense. We also see that berberine increases expression of the LDL receptor messenger RNA. Why? Because it's stabilizing the messenger RNA. And then we see a nice synergy here. We can see that berberine and low-dose statin works as well as high-dose statin. So there's this nice synergy between berberine and statin therapy. Basically, berberine will increase the efficacy of statin therapy by 40 to 50%. Now, here we're going to look at LDL receptor expression on the cell membrane. It rises with statin, it rises with berberine, it rises the greatest amount with combination therapy. Another study of berberine statin synergy. We're going to take genetically normal male Wistar strain rats, feed them over 8 weeks a controlled diet, Purina rat chow, or a high-fat, high-carbohydrate diet. We're going to basically send them McDonald's 3 meals a day. And then we're going to randomize these high-fat, high-cholesterol-fed rats into 5 treatment groups. One group gets salt water, control substance. Another group gets simvastatin. Another group gets berberine. The fourth group gets low-dose simvastatin plus berberine. The fifth group gets high-dose simvastatin. At 30 days, we're going to sacrifice the animals, evaluate their lipid levels, hepatic, that's liver, LDL receptor messenger RNA expression, and how the liver looks under the microscope, that's hepatic histology. And you can see with the bad diet in black, we have increases in LDL, triglycerides, cholesterol, and no real change in HDL. Here in green and red is simvastatin and berberine. In blue and pink, we can see that berberine and low-dose simvastatin works as well as high-dose simvastatin. Again, berberine will increase the efficiency of lipid reduction with a statin by about 50%. Now, if we look at messenger RNA for the LDL receptor, it will rise with simvastatin, it rises with berberine, and of course there's a synergy. So we lower cholesterol in the cell, we're going to make more LDL receptors. But if we lower cholesterol in the cell with a statin and shoot SREBP into the nucleus, it's going to make more LDL receptors, but it's also going to make more HMG-CoA reductase. So when we give you a statin, we're going to increase the generation of HMG-CoA reductase. Statins poison this enzyme, and as a result, an unintended result, we make more HMG-CoA reductase. This does not happen with berberine, and the increase or with combination therapy. So in a sense, statin therapy is self-attenuating. Now, I will work with you on your cholesterol, and at first we may try diet. And if diet and exercise alone does not get the job done, I might put you on a statin. And then we repeat your labs, and your LDL is lower. You come back a year later, and your LDL is up again. Now, I used to think it was because you became complacent. Now that you're taking a drug, you don't need to watch your diet as much. Human nature being what it is. And I'm sure complacency plays a role, but also so does biochemistry, because when I give you a statin to poison HMG-CoA reductase, your body's going to respond by making more HMG-CoA reductase, and I have to give you more of a statin. So in a sense, statin therapy, while it has many virtues and benefits, in a sense is irrational because the biochemical effect is self-attenuating. Of course, we can rectify that, as we'll learn with berberine. Now, hepatic cholesterol, hepatic triglycerides, how much fat is in the liver? It rises with the bad diet. There is a reduction with simvastatin, berberine, and of course there's a synergy. Here's what the liver looks like in a normal diet. This is fatty liver. The liver doesn't look so good. It's full of yellow fat, and that is attenuated with simvastatin, berberine, or combination therapy. Now we're going to look at berberine, red yeast rice extract, polycosanol, add-on therapy in familial heterozygous hyperlipidemia. This is a genetic abnormality where you don't make LDL receptors, or it's defective, or it's absent. And if you're homozygous, your cholesterol's 1500 and you die very early in life. But if you're heterozygous, one good gene and one bad gene, you will survive into adulthood. You may have a cholesterol of 400. You're at increased risk of cardiovascular disease. So they took 30 subjects with familial heterozygous hyperlipidemia, and they're all on stable doses of maximally tolerated statin plus or minus ezetimibe, which is Zetia. Zetia works by blocking cholesterol absorption. So you're blocking cholesterol absorption with Zetia, you're blocking HMG-CoA reductase, you're poisoning the enzyme with statin, you do baseline measurements. Now you're going to add in berberine, 500 milligrams a day, not a high dose, red yeast rice extract, a really wimpy dose, and polycosanol. And you're going to repeat the baseline measurements at 3 months. You see with maximal drug therapy, statin and Zetia, cholesterol fell from 384 to 245. If you add in our nutritional cocktail, it falls to 214. LDL dropped from 305 to 173 with drug therapy, drops 30 more points with the nutritional cocktail. HDL cholesterol is unchanged, triglycerides fall further, percent reduction of cholesterol, 36% with drug therapy, 44% with add-on nutritional therapy. Similar situation with LDL and triglycerides. Certainly a nice synergy here between berberine and pharmaceutical intervention. Now, this is what's kind of interesting. The greater the percentage reduction in your LDL with statins, reflecting the greater the dose of statin, the less benefit you received with a single 500 milligram dose of berberine. Now, why would that be? Why would it be that the greater the dose of statin you're taking would kind of attenuate the benefits of berberine? And as Lucy told Mr. Timmis, it's all about PCSK9. Okay, so SREBP mediates PCSK9 as well as LDL receptor transcription. PCSK9 upregulation rises with statin dosing. Berberine selectively inhibits PCSK9 transcription. So the greater your dose of statin, the more PCSK9 you're going to make, the greater will your need be for berberine. Stated otherwise, the greater your dose of statin, the greater the dose of berberine that I should treat you. Your therapeutic need for berberine will rise in relation to your statin dose because statins increase PCSK9. One of berberine's benefits is it blocks transcription of PCSK9. So here we're going to take our HepG2 cells, measure LDL receptor messenger RNA, and it will rise in a dose and time-dependent fashion with berberine. And it works by stabilizing the messenger RNA for the LDL receptor. PCSK9 transcription in a time and dose-related fashion is blunted with berberine. We see far less PCSK9 within the cell, within the circulation with PCSK9. Berberine does not affect the messenger RNA for PCSK9, it blocks its transcription. What berberine does, it enters the nucleus, it will sit on the promoter site and block reading of the genetic code for PCSK9. If we look at PCSK9 promoter activity, it is blunted. So berberine works by physiologically downregulating H-N-CoA reductase via Mother Nature's pathway, by stabilizing the messenger RNA for the cellular receptor, and by blocking PCSK9. Thus the synergy. So if we look at LDL receptor messenger RNA, it will rise with statin therapy, it rises with berberine therapy, there's a synergy. Conversely, PCSK9, something we don't want, rises with statin therapy, but it falls with berberine therapy, and the rise in PCSK9 in response to statin therapy, which is deleterious and limiting your lipid-lowering results with statin therapy, is blended with berberine. Thus, there's this nice synergy. You'll see more LDL receptor protein on the liver cell membrane with statin, with berberine, and even more with combination therapy. So this is sort of a tedious slide here, but we know with a low cholesterol diet, there's going to be less cholesterol in the liver, so sterol regulatory element binding protein translocates to the nucleus, and we're going to make more LDL receptors, and that will lower serum cholesterol. Now, the body may compensate a little bit here by making more HMG-CoA reductase and making more PCSK9, but in general, the increase in LDL receptor expression outweighs the increase in PCSK9 and HMG-CoA reductase, and our serum cholesterol falls. And of course, there's no toxicity associated with a low cholesterol diet, and there's no insulin resistance. Statins, and to a lesser extent, our green statins, red yeast rice extract and polycosanol, what they do, they block HMG-CoA reductase activity. They're really going to knock out cholesterol synthesis. Now, as an unintended consequence, they're going to make more HMG-CoA reductase and make more PCSK9, but the ability to poison the enzymes that generate cholesterol wins, and we make more LDL receptors, and our serum cholesterol falls. And of course, when we're poisoning one of Mother Nature's enzymes, toxicity occurs. We have muscle aching, cognitive decline, reductions in testosterone, reductions in vitamin D. So there's toxicity associated with statins. They're a double-edged sword, and statin therapy produces insulin insensitivity. If you take a statin, you're more likely to become diabetic as opposed to if you don't. Ezetimibe really mimics diet. It blocks the absorption of cholesterol. Now, berberine works differently. It does not poison HMG-CoA reductase. It physiologically downregulates it, and you're going to make more LDL receptors. You're not going to transcribe more LDL receptors, but you stabilize the messenger RNA, they make it a cell membrane, and they're going to pull cholesterol out of the blood, and your serum cholesterol will fall. There's no toxicity associated with berberine, and it improves insulin resistance. So it treats hyperlipidemia and diabetes and insulin sensitivity at the same time. Berberine, red yeast rice, and polycosanol versus ezetimibe therapy. 228 hyperlipidemic subjects with intolerance to statin therapy, or they refused statins. None were diabetic, none with known cardiovascular disease. So this was a primary prevention study. You put them on a low cholesterol, low saturated fat diet for 3 months. Do baseline measurements, randomize them to receive berberine, 500 milligrams a day, not a high dose, red yeast rice, 200 milligrams, a very low dose, and polycosanol, or Zetia, which acts like diet because it blocks cholesterol absorption from the gut to the liver. You're going to repeat the baseline measurements at 6 months. 14 of the ezetimibe group stopped, 8 for poor compliance, 6 to GI side effects, none on nutraceutical therapy stopped or experienced side effects. So the nutritional therapy was quite well tolerated. Zetia is not well tolerated. Cholesterol fell in both groups. LDL cholesterol fell in both groups. Nutritional therapy worked as well as did ezetimibe. No real change in HDL. Triglycerides fell in both groups, maybe a little bit more with nutritional therapy. Percent reduction in cholesterol, 19% with Zetia, 24% with nutritional therapy. LDL drops 25% versus 32% with nutritional therapy. Triglycerides fall further with the combination of berberine, red yeast rice extract, and polycosanol, which is better tolerated and less expensive. 26 of the subjects had limited response to monotherapy. Their LDL reduction was below the study mean. So you're going to take new baseline measurements and then assign them to combination therapy. Nutritional therapy and Zetia. Is there going to be a synergy? I think you know enough now to predict, yes, there's going to be a synergy. Let's go through this study. Cholesterol fell with monotherapy. With combination therapy, it falls further. LDL falls with monotherapy, does better with combination. No change in HDL, triglycerides fall, percent reduction is improved with combination therapy, and this makes sense because Zetia blocks cholesterol absorption of the liver. Less cholesterol in the liver, the more LDL receptors you're going to make. The more you're going to transcribe messenger RNA for the LDL receptor. Berberine will stabilize that messenger RNA for the LDL receptor, so you're going to wind up with more LDL receptors in the cell membrane and block PCSK9, which would break down the LDL receptor. So of course you would expect a synergy between berberine and Zetia and statins. Berberine will synergize with all other forms of lipid-lowering therapy. Now, this interaction, our cholesterol feedback mechanism, has to do with native LDL. Oxidized LDL, if we're making too much superoxide because NADPH oxidase is activated, we're going to alter the LDL. It's going to look like a microbe. So our negative feedback system that prevents our cells from overfilling with cholesterol will be defeated by oxidative stress. And hopefully berberine will be able to decrease oxidative stress. Likewise, with inflammation, we begin to store intracellular cholesterol as cholesterol ester into lipid droplets. The free cholesterol level falls, so we're going to make more LDL receptors. This allows our monocytes to engorge with lipids in response to inflammation. Now, if you have a real infection, you want to do this. You want your white cells to be engorged with lipids because they're going to need more cholesterol as they divide into daughter cells. You need the cholesterol for the cell membranes, but this is deleterious in humans who have atherosclerosis and diabetes. So let's look at inflammation and LDL receptor dysfunction. How does inflammation alter serum and intracellular lipid levels? We're going to take human coronary artery vascular smooth muscle cells. And most of our plaque is made up of vascular smooth muscle cells. We're going to put them in tissue culture with EDTA, which is an antioxidant. This will block LDL oxidation, so we're going to be looking at dynamics through the LDL receptor. We're going to incubate them with vehicle control substance, LDL, and if we give you LDL, you're going to saturate the cells so SCAP will sequester steroid regulatory element binding protein, so you're going to stop making LDL receptors. Or with interleukin-1 beta, which is a Th1 cytokine, it is a work product of Queen Jadis and nuclear factor kappa beta. It mimics infection. And we're going to measure vascular smooth muscle cholesterol content under these conditions. So here, vascular smooth muscle cholesterol accumulation, under controlled conditions, we're going to call that 100%. If we incubate the cells with cholesterol, we're going to see a rise in LDL, and we're going to begin to esterify it. We're going to store it as esterified cholesterol. An enzyme called ACAT will convert free cholesterol into esterified cholesterol to make lipid droplets, which we don't want. So of course, if we overfill the cells with cholesterol, they're going to store some of it. If we overfill them with cholesterol and add in inflammation, interleukin-1 beta at a low dose, we're going to increase free cholesterol a little and esterified cholesterol a lot more in a dose-related fashion. The more inflammation, the more we upregulate ACAT, the more we transfer free cholesterol into esterified cholesterol. So if we lower the amount of free cholesterol, SCAP senses this, sends SRE binding protein to the nucleus. We're going to make LDL receptors even though the cell is overfilled with cholesterol, because we're seeing only the free cholesterol. With inflammation, we transport cholesterol into esterified cholesterol. This is how inflammation leads to cellular overfilling with cholesterol. If we look at LDL receptor messenger RNA, it will drop when the cells are full of cholesterol because SCAP will sequester SREBP. But if we add inflammation and esterify the cholesterol, free cholesterol levels fall, we're going to start transcribing messenger RNA for the LDL receptor. Now, normally high cholesterol in the cell will lower production of the LDL receptor, but if we add in inflammation, we overcome the negative feedback. Interleukin-1 beta effect, inflammation effect on messenger RNA for the LDL receptor without LDL loading will increase it. Because you're going to increase esterification, you're going to make more SCAP, more SREBP, more LDL receptors. The effect with LDL loading, now if we load the cell with LDL, we're going to make less SCAP, less SREBP, less LDL receptor, but as we add inflammation, we'll make more of these inappropriately. And this allows our cells to overfill with lipids in the presence of inflammation. Same dose of cholesterol, you add in inflammation, our cells will overfill with lipid. They form cholesterol droplets, and our white cells are going to become foam cells and cause atherosclerosis. Here, we're going to look at SCAP, the Golgi apparatus, and an overlay. In cholesterol-depleted media, we will transport SCAP into the Golgi. You see the overlay here. When there's plenty of cholesterol, we're going to do that. We're not going to transport SCAP to the Golgi and then the nucleus. We stop making LDL receptors. If we add in inflammation, we'll start making LDL receptors again inappropriately if we don't want to get heart disease. But of course, this was all set up to help us fight infection. Unfortunately, our body is misinterpreting our current milieu of bad diet, leaky gut, inflammation, gingivitis, overweight, sleep apnea, toxicity as an infection. So we're making LDL receptors inappropriately. We're overfilling the cells of our body with lipids, and we're developing atherosclerosis. LDL alone, if we add in tumor necrosis factor alpha, interleukin 1 beta, any of the inflammatory mediators will cause cholesterol esterification. Inflammation leads to cholesterol esterification. Lower intracellular free cholesterol, SCAP sends SREBP into the nucleus. We're going to make more LDL receptors. Cholesterol overfilling with cholesterol. The negative feedback mechanism is overcome, which is why we're seeing heart disease in people with normal cholesterols. It's not your cholesterol level per se, it's how your body manages the cholesterol. Under inflammatory and oxidative conditions, you're going to overfill your cells with cholesterol. The oxidized LDL looks like a microbe, more inflammation, more atherosclerosis. Toll-like receptor for myeloid differentiated factor 88, crosstalk with SCAP SREBP. We're going to take human monocyte-derived macrophages. These are the cells that mediate atherosclerosis. We're going to incubate them in tissue culture with LDL, and that's going to saturate the cells, so they're going to sequester SCAP in the endoplasmic reticulum, so you're not going to make more messenger RNA for the LDL receptor. Lipopolysaccharide, which will activate the toll-like threat receptor, that will activate Queen Jadis to shoot nuclear factor kappa beta in the nucleus. LDL and lipopolysaccharide, with or without a molecule that blocks the toll-like receptor. Now, all of our cells, particularly our white cells and our liver cells, have all these threat receptors. The toll-like 4 myeloid differentiated factor 88 receptor will be ligated by bacterial lipopolysaccharide. That's the material in the cell wall. Incidentally, it's also aggravated by free fatty acids. So when it is activated, a chain reaction of serine kinases is initiated, and this will activate Queen Jadis, ICPA kinase. ICPA kinase, Queen Jadis, will degrade the inhibitor of nuclear factor kappa beta. Nuclear factor kappa beta translocates in the nucleus to make IL-6, IL-1 beta, tumor necrosis factor alpha, adhesion molecules, matrix metalloproteinases, all these molecules that mediate atherosclerosis that were given to us by evolution, our creator, to kill bacteria. Instead, they're killing our arteries. Now, so we're going to do this study with or without a molecule that will block the toll-like receptor. So if we block the toll-like receptor, we will not be able to activate ICPA kinase Queen Jadis. So we knock this out, then lipopolysaccharide or free fatty acids will not translocate nuclear factor kappa beta in the nucleus. It'll blunt trafficking through the toll-like receptor pathway. So we're going to take these human monocyte-derived macrophages, treat them in this fashion, and we're going to look at macrophage cholesterol content. So what happens here is we ligate the toll-like MyD88 receptor. We're going to shoot nuclear factor kappa beta in the nucleus, make all these pro-inflammatory cytokines. They promote cholesterol esterification, and this is the mechanism by which our negative feedback mechanism is overcome by inflammation. So without blocking the toll-like receptor, we see that we don't have much cholesterol filling of our cells on a low cholesterol diet. It will increase with lipopolysaccharide, it will increase with LDL, and there's a synergy. High LDL, high lipopolysaccharide, our cells were going to be engorged with cholesterol. If we knock out the toll-like receptor and we block the generation of inflammatory cytokines, this cholesterol overfilling in response to inflammation is dampened. So it's all going through the toll-like receptor, Queen Jadis, ICPA kinase, nuclear factor kappa beta going to the nucleus, making inflammatory mediators and bullets. That is why our cells are overfilling with cholesterol. If we look at levels of messenger RNA for the LDL receptor in the liver, if this is in our monocytes, and we'll see later the opposite occurring in the liver, with a low cholesterol media, we don't see much messenger RNA. If we add in the lipopolysaccharide, we're going to make a lot more, although if we block the toll-like receptor, if we block inflammation, that doesn't occur. If we incubate the cells with LDL, they make less messenger RNA for the LDL receptor, but the suppression in the transcription of the LDL receptor with high cholesterol is blunted by inflammation, allowing the cellular overfilling. If we look at levels of HMG-CoA reductase, they are increased with lipopolysaccharide, they're decreased with LDL. That makes sense. If you got plenty of LDL, you don't need to make more, but with inflammation, you're going to lower the free cholesterol because you're esterifying it. The body thinks there's not enough cholesterol in the monocytes, you're going to make more. So our negative feedback mechanism is blunted. And these more pretty pictures here showing that in the presence of inflammation, SCAP is going to take SREBP into the nucleus to make more LDL receptors, even though the cell has plenty of cholesterol. And this is the explanation as to why inflammation leads to heart disease in people with normal cholesterols because of this crosstalk. So threat ligates the toll-like receptor, Queen Jadis, ICPA kinase is activated, she shoots nuclear factor kappa beta in the nucleus, making cytokines and other bullets. We begin to esterify our cholesterol, less free cholesterol in the cell. SCAP will take SREBP into the nucleus. We're going to make more HMG-CoA reductase, more LDL receptors. We're going to overfill our cells with lipids. We get foam cells and atherosclerosis. Inflammation affects on cholesterol metabolism. In the liver, with inflammation, we are going to make fewer LDL receptors. The liver cells will take up less cholesterol. They make more cholesterol. They make more PCSK9. In inflammatory conditions, the liver becomes a net producer of cholesterol. It's making cholesterol to meet the needs of the body to help it fight infection. In monocytes, in vascular smooth muscle cells, mesangial cells, cells of the kidney. So far, all the cells of the periphery, with inflammation, they become net cholesterol importers. With inflammation, we're going to transfer cholesterol from the liver into the cells of the body, particularly the white cells, and this mediates atherosclerosis and its consequences. Now, this is appropriate if you got a real infection, but if you have the pseudo-infection, the milieu of modern Western man, this is mediating hyperlipidemia, hyperglycemia, endothelial dysfunction, atherosclerosis, and its complications. This decision to upregulate cholesterol production in response to perceived infection leads to oxidative stress and inflammatory stress. We are going to oxidize our LDL. We're going to shoot deleterious transcription factors in the nucleus and make cytokines causing cellular overfilling with lipid. Now, antioxidant and anti-inflammatory therapy. Can berberine blunt oxidative stress, block LDL oxidation, blunt inflammatory stress, blunt cholesterol overfilling of our white cells? Could berberine blunt hyperlipidemia in response to inflammation, blunt hyperglycemia in response to inflammation? Now, when we upregulate cholesterol production in response to perceived infection, we're going to generate a class of molecules called isoprenoids. And they are signaling molecules to upregulate inflammatory pathways, particularly Rho kinase. Rho kinase upregulates NADPH oxidase, which makes superoxide. This is the most important free radical in the body. The more superoxide we have, the more free radical stress, more degenerative disease. So we're going to take human macrophages, white cells in tissue culture. We're going to pre-incubate them for 20 minutes with vehicle, controlled substance, or berberine. Then we're going to stimulate the macrophages with lipopolysaccharide over increasing time periods. Lipopolysaccharide will bind the toll-like receptor. Queen Jadis will shoot nuclear factor kappa beta in the nucleus. We're going to make a lot of inflammatory mediators, more oxidative stress. So in response to lipopolysaccharide, we're going to make a lot of superoxide. That's produced by NADPH oxidase, which is upregulated inflammation. And berberine does not affect this. But at 6 hours, under control conditions, we're still cranking out too much superoxide. This is blunted by berberine. Now, this is important. In response to acute infection, you want your body to make bullets. We don't want berberine to block this, otherwise berberine would prevent us from responding appropriately to infection. We want to respond to infection, we want to stand down. And berberine allows us to stand down and stop making free radicals inappropriately. Human macrophages in tissue culture pre-incubate for 20 minutes with vehicle or berberine at increasing concentrations, stimulate the macrophages with a fixed dose of lipopolysaccharide over a fixed time period. So here we're looking at superoxide under control conditions. Berberine has no effect under control situations. Lipopolysaccharide increases superoxide generation, and this is blunted in a dose-related fashion by berberine. High-dose berberine blocks the generation of superoxide below that of control conditions. Now we're going to take the cells and incubate them with vehicle, berberine, or apocinin, which is an artificial NADPH oxidase inhibitor. It will poison the enzyme. Then we're going to stimulate the macrophage with lipopolysaccharide. And you see an uptick in free radical generation with lipopolysaccharide that is blunted by berberine. It's blocked by apocinin, which blocks NADPH oxidase. NADPH oxidase is upregulated by lipopolysaccharide. That makes sense. It responds to perceived infection. It is blocked by berberine. It is blocked by apocinin. This is telling us that berberine blocks the activation of NADPH oxidase. It will restrain NADPH oxidase, blunting inappropriate generation of superoxide. Now, so NADPH oxidase is such a powerful generator of free radicals that its components are kept apart within the cell. In response to ligation of a threatening molecule with the receptor, like the toll-like receptor in the cell membrane, Rho kinase will activate NADPH oxidase, and the parts come together. And so if you add in lipopolysaccharide, you'll see an increase in this particle called P22. Berberine has no effect. Berberine had no effect on particle 22, 67, 47, or RAC, but it did knock down particle 91. Lipopolysaccharide will increase this. Berberine blocks it. Particle 91 is the gatekeeper for NADPH oxidase. So by blocking particle 91, we're going to downregulate NADPH oxidase and blunt inappropriate or prolonged superoxide generation from NADPH oxidase, which is of great value in preventing cardiovascular disease and any degenerative disease. Now, atherosclerosis begins when lipid particles infiltrate the artery wall. Now, LDL is food, and if it's not needed, if it's not recognized by an LDL receptor, it'll swim out of the artery wall unless the particles get stuck. And what happens is that phospholipase enzymes, which are upregulated by inflammation and oxidative stress, will alter the lipid particles. It'll oxidize the phospholipids in the LDL cell membrane, and they stick together. Then they get stuck in the artery wall. They're acted upon by free radicals. The LDL is oxidized. The oxidized LDL is misrecognized as a microbe. White cells infiltrate the artery wall, and this initiates and propagates atherosclerosis. So the initial mistake is modification of the LDL by oxidation of its phospholipids. So they stick together into these clumps. And then you get this clump of LDL that is stuck to the ground substance of the artery wall. It can't swim out anymore. The modified LDL is stuck. It can be acted upon by free radicals. Then it'll be mistaken as a microbe. So let's look at the effect of berberine on LDL oxidation and endotheliatoxicity. We're going to incubate LDL with copper, and copper will oxidize the LDL. And we're going to measure malondialdehyde. That is a marker of oxidative stress at the level of the phospholipids. So for your LDL to get stuck in the artery wall, the phospholipids are modified, they're oxidized, and malondialdehyde would be a marker of that. And you can see with copper, we make a lot of malondialdehyde, and we're blunting this with berberine in a dose-related fashion. We also blunt it with Trolox, which is an antioxidant. Berberine and Trolox will protect against malondialdehyde formation, and this protects the LDL from phospholipid oxidation, and we can extrapolate this and say that it will prevent cell membranes from being oxidized. All right, so we're going to incubate the LDL with copper, and now we're going to measure oxidized LDL. We do not want to oxidize our LDL because it'll be mistaken as a microbe. And this occurs with copper, and we can block this with berberine and Trolox. Berberine and the antioxidant Trolox protect against LDL oxidation. Now here we're going to look at fragmented ApoB. The LDL has been modified, it's been oxidized, the protein has been altered, and it now looks like a microbe. And we're going to measure ApoB fragmentation. This occurs with oxidative stress, and both berberine and Prolox protect against ApoB fragmentation. So is berberine an antioxidant? Now, we're going to do this study in cell-free media. This is just a chemical reaction in a test tube. And we're going to measure the ability of berberine and Trolox to scavenge free radicals. And as you can see, berberine is a very ineffective antioxidant. It will not directly neutralize free radicals. Trolox is. So Trolox is a strong antioxidant. Berberine is a weak antioxidant in cell-free conditions. Therefore, it must be working by stimulating enzymes within the cells to provide antioxidant function. Now, if we incubate human umbilical vein endothelial cells, these are umbilical vein endothelial cells obtained from newborns, and they are used in cell culture studies. And you incubate them with oxidized LDL, you're going to damage the cells. They commit apoptosis, they commit cell suicide, because oxidized LDL is toxic to endothelial cells. But if you add in berberine or Trolox, you're going to prevent that. So berberine and Trolox protect against oxidized LDL-induced cytotoxicity of the endothelial cells. Here we're going to look at apoptosis. The colored cells, the nucleus has basically died and they're committing cell suicide. This is damage to the DNA. Berberine and Trolox blunt oxidized LDL-induced, free radical formation-induced damage of the nucleus and cell suicide apoptosis. We will be talking a lot about apoptosis in a subsequent lecture, but what happens is we get all this free radical stress, we're not making enough energy, the mitochondria begin to fail, the electrochemical gradient across the membrane of the mitochondria is lost, molecules that are supposed to be in the mitochondria enter the cell fluid and vice versa, and the cell begins to commit suicide, apoptosis, and this is blocked by berberine. So the toxicity, cell suicide tendency of oxidized LDL on endothelial cells is blocked by berberine. We have these apoptosis pathways, which we'll discuss later. This is blocked by berberine. Berberine provides not a direct antioxidant effect. It's not like vitamin E or vitamin C. It will not squelch a free radical, but what it does, it works with our enzymes. Berberine blocks NADPH oxidase. So it's not an antioxidant, it blocks the generation of free radicals. If we block the generation of superoxide, there's no free radical stress. And on DVD 2, we'll talk about how berberine promotes translocation of NRF2 into the nucleus. NRF2 is a beneficial transcription factor. It codes for antioxidant enzymes like superoxide dismutase and catalase. So it upregulates our antioxidant defenses and promotes cell repair and neutralizes toxins. So berberine is working indirectly by enhancing our physiology in a favorable fashion. Now let's look at berberine in inflammation, lipopolysaccharide, pseudo-infection-induced dyslipidemia. 40 genetically normal female mice, randomized them to receive over 4 weeks, chow diet, chow with berberine low dose, chow with berberine high dose. After the 5th week, the 3 interventional group will receive a high cholesterol diet over 7 days. Then you're going to treat them with intraperitoneal lipopolysaccharide to mimic surgical sepsis, peritonitis. And we're going to look at plasma 8 isoprostane, which is a marker of oxidative stress. So in response to this artificial peritoneal sepsis, with lipopolysaccharide, we see free radical stress in the circulation, which is blunted by berberine. So we're providing an antioxidant effect. Lipopolysaccharide will bind the toll-like receptor, and that will shoot nuclear factor kappa beta into the nucleus and make transcription factors. In a subsequent discussion, we're going to show that berberine blocks the toll-like receptor and it blocks Queen Jadis. So it's going to blunt inflammation in response to real infection or perceived infection. So in response to lipopolysaccharide injection of the peritoneum, reflecting surgical sepsis or peritonitis, we see upticks in inflammatory mediators like tumor necrosis factor alpha, interleukin-1 alpha, interferon gamma, that is blunted by berberine. If we look at LDL receptor protein expression in the liver, it is decreased. We make less messenger RNA, we make fewer LDL receptors. This is blunted by berberine. So with inflammation in the periphery, we make more LDL receptors. In the liver, we make less. So we're not pulling cholesterol out of the circulation of the liver. The liver becomes a cholesterol generator, puts it into the circulation. It's not going to pull it back out because it's all going to go into the periphery, into the mononuclear cells. PCSK9 that degrades the LDL receptor and raises cholesterol, in the presence of inflammation, the liver's going to make more of it. So you're going to get hyperlipidemia. Berberine blocks this. So these deleterious changes, producing a net transport of cholesterol from the liver into the periphery in response to inflammation, is blunted by berberine. So with inflammation, you see upticks in cholesterol, LDL, and triglycerides that are decreased by berberine. Inflammation shunts cholesterol from the liver to the periphery, and we block that with berberine. The decision to increase cholesterol production is a misdecision. It's a mistake. Our body is misperceiving our high sugar and our high fat diet, our belly fat, environmental toxins, leaky gut, infection, allergy, gingivitis, any sort of inflammation as another infection. And we spent the last 3 million years getting good at handling infection. We are going to activate inflammatory pathways. We're going to activate Queen Jadis. She's going to shoot nuclear factor kappa beta into the nucleus, and we're going to respond to this perceived infectious threat with more cholesterol, more sugar, more bullets, endothelial activation, leading to hyperlipidemia, diabetes, and atherosclerosis. Berberine fulfills Pohling's postulates. So far, we've covered the role of berberine in dealing with lipids, dealing with oxidative stress, and inflammation to some degree. We've defined the mechanism of action of berberine at the levels of transcription and translation. Berberine has limited toxicity. It costs $30 a month. And it synergizes with pharmaceutical therapies, and we'll show later on that it improves outcome following stent placement. And we have published studies to document mechanisms and efficacy. Hepatic cholesterol metabolism. Sterol regulatory element is the promoter site for transcription of the LDL receptor. Sterol regulatory element binding protein binds to the SRE. SCAP keeps it in abeyance. When we have plenty of cholesterol in the cell, SCAP sequesters SREBP. We're not going to make HMG CoA reductase. We're not going to make LDL receptors. When cholesterol is low due to starvation or due to drug therapy, SCAP senses this. It will transport SREBP from the endoplasmic reticulum to the Golgi apparatus into the nucleus. We're going to make more LDL receptors. That's the negative feedback system that was designed for primitive man. Berberine activates AMP-sensitive protein kinase. We want you all to follow the AMP-sensitive protein kinase lifestyle. We want you to exercise, put down the knife and fork at the appropriate time. We don't want you to suffer from an excess of inflammatory cytokines or an excess of energy, because that will turn off AMPK. We want you to exercise. We want you to remain trim. And we, of course, want you to take berberine to upregulate AMP-sensitive protein kinase, because when we do this, we physiologically downregulate the production of cholesterol and triglycerides. In our next section, we'll talk about the role of berberine in treating insulin sensitivity and in weight control. And we'll talk about its effects on oxidative stress and inflammation and protection against toxicity and malignancy. Oxidative stress is inflammatory stress. The misdecision to make cholesterol activates NADPH oxidase, lots of free radicals. We're going to open up our inflammatory pathways. We're going to oxidize our lipids. We're going to overfill our cells with lipids. We're going to get atherosclerosis. The physiologic effects of berberine all follow activation of AMP-sensitive protein kinase. We talked about lipid metabolism. We're going to physiologically downregulate HMG-CoA reductase so you make less cholesterol. If we have less cholesterol, we're going to increase transcription of the messenger RNA for the LDL receptor, and we're going to stabilize it. We're going to block PCSK9. We're going to see decrease in serum cholesterol. Likewise, we will downregulate the enzymes that generate fatty acids and triglycerides. In our next section, we'll talk about insulin receptor expression and insulin signaling. We have a beneficial effect on weight. We've talked about the role of berberine in blocking oxidative and inflammatory stress, primarily by restraining NADPH oxidase, blunting inappropriate generation of superoxide. We improve endothelial function, we improve cardiovascular physiology, and we lower inflammation. Clinical effects, LDL and triglycerides fall with minimal change in HDL. Hyperlipidemia due to inflammatory and oxidative stress will be attenuated. Glucose falls. Hyperglycemia in response to inflammation falls. Weight loss may occur. Fatty liver improves. We protect against cellular damage due to infection, inflammation, insufficient oxygen, or toxins. Protection against malignancy based on animal studies. Tolerance to radiation therapy improves. Endothelial function improves. BP may fall. We protect the heart from stress. There's protection, at least in animal models, against restenosis. We improve outcome following stent placement. There's benefits in heart failure. Berberine synergizes with and adds to standard pharmaceutical measures. How do we use berberine? The standard dose is 500 milligrams twice a day, lunch and dinner, or breakfast and lunch. We could advance to TID 3 times a day if the result's suboptimal, or if you're on high-dose statin therapy, you're going to need more. There's a synergy between berberine and statins and red yeast rice and polycosanol and Zetia. 50% reduction in statin dose is likely possible. So if we need you on a statin because your cholesterol is too high and you're having side effects, I can add in berberine, cut back on your statin dose, get the best of both worlds, good cholesterol reduction, good reduction in inflammation, improvement in blood sugar, fewer side effects. What are the downsides of berberine? Not a whole lot. 5% of you will experience nuisance GI side effects, constipation or diarrhea, and all you have to do is decrease the dose. This is a dose-related side effect. If you are on insulin or sulfonylurea drugs that kick insulin out of the pancreas, and we improve insulin sensitivity with berberine, we're going to have to cut back on your insulin dose, otherwise you might get hypoglycemic. There's a single case report in the world literature of reversible bradycardia, that's low heart rate, with the use of berberine that we'll discuss in disc 2. So it's very, very safe. We can't really hurt you with berberine. So we entered the world of berberine through the wardrobe. We took the path by the light post, and we've learned a great deal. And now we can return to our everyday lives and retain and use this knowledge to do good. And then we will sail forward to the next section, berberine and diabetes management and weight control, which is really titled Berberine and the Evil Doers of Insulin Resistance, Cytokines, Free Fatty Acids, and IC kinase.